The role of DNA ligase in DNA replication is to option A. Add more nucleotides to the growing strand one at a time. Option B. Ligate base to sugar to phosphate in a nucleotide. Option C. Join Okazaki fragments to one another. Or option D. Remove incorrectly paired bases. So, the correct answer would be option C. During replication, the Latin strand is synthesized in short, discontinuous segments known as Okazaki fragments. DNA ligase seals the mix between these fragments by forming phosphodiester bond between the 3' hydroxyl end of one fragment and 5' phosphate end of the adjacent fragment, creating a continuous strand of DNA.